Okay, so today I'm going to be talking to you about microbial natural products, and I'm going to be covering a few topics: drug discovery, metabolomics, and chemical ecology. Normally, you can find me in room C5125, but I'm not in China at the moment. I'm stuck in New Zealand. So I'm going to start by discussing what is a natural product. And to do that, we need to divide small molecules into two classes. Within a living organism, we have primary metabolites, and these are compounds or small molecules that are crucial for the growth and normal development of an organism. So these should be things you're familiar with: amino acids, sugars, nucleic acids, proteins, and fats. So secondary metabolites are compounds produced by living organisms that aren't primary metabolites. They often provide a selective advantage. They could be antibiotics, they might be used in signaling or something like that. And they're often slightly different in their structure from primary metabolites. So on the bottom left, we have some examples of primary metabolites. This is glucose, and here is an amino acid, alanine. And then on the right, we have some secondary metabolites, a terpene and tetracycline there. And these secondary metabolites often have useful pharmacological properties, so they can be used as drugs. So natural products are great. They have unique structures that you don't find in synthetic compounds. And in fact, they have quite a good record of being used as drugs. So over the period 1981 to 2010, natural products, their derivatives and mimics comprise 59% of all small molecule drugs. And if you look at the graph, we can divide this into real natural products, natural product derivatives, synthetic compounds with a natural product pharmacophore, or compounds that have been designed from the knowledge of a natural product. So that's quite a significant portion. Now, why else would you want to study this field? Well, maybe you could win a Nobel Prize. Maybe. So a few years ago in 2015, Tu Youyou was one of the people who won the Nobel Prize in Medicine for her discoveries on antiparasitic natural products research. And she won that in conjunction with two other researchers, William Campbell and Satoshi Omu, for natural product discoveries, also for antiparasitic compounds. But for me, these people are not the real heroes. The real heroes are the molecules, artemisinin and ivermectin. And you can see these have some pretty interesting structures. So when it comes to natural product, I showed you before, they have some slightly different structural classes compared to primary metabolites. So these different classes are made using different processes from different components. So a polyketide, for instance, is made out of acetate. So here's acetate here. And here's an example. This is doxorubicin, which is an anti-cancer compound. We have terpenes made from isoprene units. Peptides made from amino acids, or alkaloids, which are also derived from amino acids. So this compound here, for example. And many of these are useful drugs. We can get natural products from many different sources. Medicinal plants have a very long history in human health. Many of you will probably be familiar with the traditional Chinese medicine, but there are other traditions around the world. And this can be very useful for drug discovery. trying to find pure compounds from mixtures or preparations that have been used traditionally for hundreds or thousands of years. The first pure substances were purified usually in the mid-19th century, so 1850 or so. Some examples include vincristine, an anti-cancer alkaloid from the plant we see on the left, or cannabidiol, which is from Cannabis sativa, and this is used to treat epilepsy. Another potential source of natural products are marine natural products. These have only recently been investigated for their chemical potential. Most notable success is from marine invertebrates, sponges, corals, but there is a limited supply often for these compounds, and environmental damage can be a problem. You can't go and collect every single sponge that you find because that could be devastating to the ecology of these organisms. In the marine environment, we see highly unusual structures not found in terrestrial organisms. Cytarabine is an anti-cancer agent derived from compounds from a marine sponge, and trabadectin on the right is isolated from a marine tunicate and is used for cancer treatment. But the ones that I'm most interested in are microbial natural products. These are medicinal compounds found in bacteria and fungi. Molds and macrofungi have been used to treat wounds for many centuries, but again, the discovery of the pure substances only began within the past 100, 150 years. Why do I like microbes? They have wonderful structural diversity. We can see down here many, many different types of compound with different types of structure. You can easily alter metabolite production, and you can grow the microbes again. So these are some of the microbes I've played with in my lab. These have been isolated actually from the Tianjin University campus. A little bit more history. So one of the classes of microbial natural products you should be familiar with are the penicillins. These were first discovered in 1928 by Alexander Fleming, and it's rather a famous story. He had a very dirty office, and he eventually found that one of the plates where he was growing his bacteria, the bacterium had died in the area surrounding a fungal contaminant, and that led a lot later to the discovery and purification of penicillin. Industrial production of the compound actually began in 1943, and this was during the Second World War, so having a good, reliable antibiotic was very, very useful when treating soldiers who might get infections. It is a potent broad-spectrum antibiotic, and we can see Alexander Fleming here, and here's one of the most well-known penicillins, penicillin G. Another class are the statins. Now, the statins are used in the treatment of high blood pressure. They lower cholesterol levels in the blood. Mevastatin was discovered in the 1970s by a Japanese company, and we see this over here. It's found in red yeast rice. And lovastatin, another similar molecule here, has been sold commercially from 1987 as Mevacor. So they inhibit HMG-CoA reductase and lower cholesterol levels. So I'd just like to ask at this stage, are there any questions that any of you have for me? No? Okay. So that's natural products in general. I'm now going to move on to discuss my background a little bit. I come from New Zealand, this little country down here, and I did my undergraduate degree at Victoria University of Wellington, majoring in chemistry and biochemistry. I then moved just a little way to Australia, to Brisbane, to do my PhD in microbial natural products. I then did a bunch of postdoctoral stints in Scripps Institution of Oceanography in San Diego, in the University College Dublin in Ireland, the University of Hawaii, where I studied the metabolomics of Hawaiian organisms, and my postdoctoral supervisor there was actually Professor Bob Boris, who is faculty here at TJU. And then I moved to my first faculty position at Tianjin University. So my group focuses on microbial natural products drug discovery. I have 5 postgraduate students, 1 postdoc, and we're a friendly group with a cooperative attitude. And my projects mostly focus on drug discovery, chemical ecology, and metabolomics. I'm going to start by discussing drug discovery. So China has a long history of natural products research, but this is mostly associated with the traditional Chinese medicine. And traditional Chinese medicine is mostly plants, a few macrofungi, and a few marine organisms, but not many. So the microbial diversity in China is relatively poorly investigated. China is a huge country. It has diverse environments, and diverse environments can produce diverse microorganisms, which gives us lots of different chemicals. So here are some of the places I have collected. I've collected the desert near Turpan, in the Yellow Mountains, in Laolongtou, and also just very close to my lab in Peiyang Square. So once we get a microbe, the first thing we want to do is make sure we have the maximum number of compounds from one microbe. So we can change the culture conditions, we can change the temperature, the media, the salt content, and we can add specific chemical agents to change the way metabolites, natural products, are being produced. You can add cytochrome P450 inhibitors or antibiotics in small concentrations and get new compounds. And for fungi, you can add epigenetic modifiers, which change the binding and activation of DNA. And here's an example from the literature. If we grow the fungus under normal conditions, it's white, and the HPLC looks very, very boring. If we add an epigenetic modifier, then suddenly it turns bright orange, and we see many, many, many new compounds being produced, like this one here. Once you have grown your microbe, then you want to isolate the compounds. And usually we start big and finish small. So we would start by extracting the microbe. Then we would carry out liquid-liquid chromatography, then open column chromatography, and usually we will finish with HPLC. Once we have isolated the compounds, we need to determine the structures, and we do that using spectroscopy. We can use mass spectrometry to determine the molecular mass. This is a very sensitive technique. We can then use nuclear magnetic resonance, NMR, to give us detailed structural information using 1- and 2-dimensional techniques. And we can also use other techniques such as infrared spectroscopy UV vis, circular dichroism, and other things. All of these give important information. So I'm just going to take you very quickly through how a structural elucidation might go. We start off with a mass spectrum, and from the mass spectrum, we can determine a molecular formula. Then we run our proton NMR. Our proton NMR gives us some complex signals that tell us about the different parts of the molecule. Where are the different protons? What is their chemical environment? And using this information, we can find several different pieces of the molecule. Once we have the pieces, we use two-dimensional NMR to put those pieces together. Here is the complete molecule, and we see here some of the key correlations. So the correlation between a proton and a carbon. So it goes from this proton here to this carbon, so we can connect them here. We can then look for other correlations to connect the rest of the molecule together. And finally, we can go back to the MS this time, and we look at these fragment ions. So this is your molecular ion, and here is a fragment ion. And you can see the fragment comes from cutting the molecule in this position. And we have other fragments we can form by cutting the molecule up in different places. So this helps to confirm our structure. Once we have our compounds, we've determined the structures, then maybe we can make some analogues, some derivatives. Natural products have complex scaffolds, and if we can alter them, we can change the potency, the selectivity, the solubility, and maybe the metabolism. Possible methodologies include semi-synthesis, enzymatic methods, or biosynthetic methods. So here we have a compound, amoxicillin. This part here comes from a microbe, and this part here is added later using a synthetic method. Here's another example. This is a natural product. It's too toxic to use as an antibiotic, but if you make a derivative like this, then you have a useful antibiotic that can be used on your skin, and this was developed recently by GlaxoSmithKline. So I'm going to give you two examples of isolating natural products and then making derivatives. So this is an old example from my own work many, many years ago. So we had a fungus, and you can see the fungus here has a lovely yellow-orange color. And this gave us three new compounds, three new polyenes. One new pyrrole, so the pyrrole, and two furans here and here. This compound had potent cytotoxic properties. So you can see the LD99 is very low. So this is submicromolar. These ones, not so good. So we had a nice natural product with good activity. We published the paper, and then we tried to see, can we make any more derivatives? So we used a technique called Precursor directed biosynthesis, where you use a modified starter unit to produce a modified natural product. And we carried out feeding studies with some pyrrole carboxylic acids. So I made or purchased many of these compounds, and then you feed them to the microbe, to the fungus. And it turns out the fungus can accept these pyrroles here and use them to produce new compounds. So here we have chloro, bromo, and fluoro carboxylic acids incorporated successfully. So you can see here, this is an HPLC. This is normal conditions. When we add the chloro compound, we see a new peak, and the new peak is this new compound. When we add the brominated compound, we see a new peak. This is a new brominated compound. And the same when we add a new fluorinated compound, we get this new natural product derivative. And if we look at the anti-cancer activity, we find that the brominated compound has very good activity and different cell activity. So that's a very nice result. Here is a newer story, and this is one from my research here in Tianjin University. So one of my students, Shi Yue, she worked on this microbe here, which develops a nice yellow color as you grow it. It has an interesting HPLC, which is dominated by these two huge peaks. She did a growth, separation, and she got these compounds here, including 2 new compounds. And we published that. That was nice. But we also had huge amounts, hundreds and hundreds of milligrams of these compounds here. So what can we do with that? Well, we gave those to another student, Li Jianye, and what he did is he took the natural product here and he made new derivatives, made 23 new derivatives. And some of these had improved activity. And in fact, one of them, this compound here, had much, much better activity. So then the next step was to figure out what is happening with these compounds. How does the activity work? So we did some electron microscopy. So here is the control microbe. This is Staphylococcus epidermidis. And see here, we have these nice round balls. It's looking very happy, very healthy. But if we treat it with the compound, we can start to see many of the cells are dying. They have damage to their membranes. So this might be acting by hitting the membranes. We can also run time course experiments, and these show at a higher concentration, we get very rapid death of the bacteria over about 10 to 24 hours. So if you do drug discovery project, what would you do? You'll learn useful techniques. HPLC, LC-mass, NMR are all useful in industry and academia. You could do some simple synthetic chemistry. You can solve some puzzles. I like solving structures. It's a lot of fun. And you can publish some papers in international journals. I'm going to speak now briefly about metabolomics. Metabolomics is the study of the total metabolite content of an organism. And this involves identifying the compounds, quantifying the compounds, and doing some analysis. So we could identify markers that are different between two groups. So that could be diseased tissue versus normal tissue, could be different species or varieties. And then we can try and classify the organisms or samples. So here's an example from my own work. These are some plants called anthuriums, and we were looking at the flavonoid constituents. And are there different chemical differences between the types? And you can see here, they have different colors and different shapes. And the HPLC profile here changes depending on the color and the shape. And what we can do is use this to sort the samples by carrying out a principal component analysis. And we can see that these heart-shaped red ones over here have a certain chemical makeup, whereas these thinner tulip-shaped ones have a much more diverse chemical composition. Using that information, we can again go to the loadings, and each of these dots is a compound. And the compounds over here are associated with this type, and the compounds over here are associated with this type. And by doing that, we can figure out we have different compounds associated with different ancestries of the various hybrids. We can also look for stress markers. So we can look at the monthly variation in the natural products of a plant. And some of the compounds go up and down depending on the temperature, like this. And some compounds go down and up depending on the temperature. And so I'm still looking into finding what are those compounds and what do they do. So metabolomics plus drug discovery allows several powerful analyses. We can categorize things into multiple chemotypes. We can rapidly identify known natural products, and we can make a correlation between metabolites and activity. So you'll learn useful techniques. If you do metabolomics, however, you'll have to do some mathematics. You'll use statistical techniques and packages such as R, XCMS, etc. Omics, metabolomics is very, very fashionable right now. So there is a potential to get some nice high impact publications. The bad point is you do need to use mathematics, and I know a lot of students don't like that very much. I'm going to conclude by talking about chemical ecology, which is the study of chemicals involved in the interactions of living organisms. How do microbes interact with each other? How do endophytes and pathogens interact with the host? So here's an example of an ecological interaction. This is biofilm formation, and this is where the microbes, when they settle on a surface and there is enough of them, they start to produce a biofilm, and that biofilm protects them from antibiotics and from other microorganisms. And what causes them to make this biofilm? It's this compound here. When this compound reaches a high concentration, they start to produce a biofilm. And you can take a related compound and feed it to them, and that will destroy the biofilm. You can also see microbes, when they interact at these cross points, they start to produce different pigments in this instance. So we can see a yellow fluorescent pigment being produced at the interaction points. One thing we can investigate is microbial co-culture. Small molecules can mediate microbial interactions. So can growing microbes together make new compounds? Well, the answer is yes. So this is one of my students, Mohammed Salim, and he found this is one fungus here growing by itself. This is another fungus here growing by itself. But when you grow them together, you get this bright red pigment being formed. And if we look at the HPLC, we see the profiles are different, but if we grow them together, we get these big spikes of new compounds being produced in co-culture. And we're currently investigating what those are. We can do something similar with plants. Many microbes live inside plants. They're often called endophytes. How do the microbe and the host interact with each other? And are small molecules affecting or affected by the interactions? So here's a similar study. We have a plant. this is the HPLC. We have a fungus that we have isolated from the plant. And here is the HPLC of this one. And then if we grow the fungus in the presence of the plant extract, we see a huge change in the HPLC. So we see these new compounds being produced, and this compound here suddenly disappears. So again, we're isolating those compounds and seeing exactly what's going on. So if you do a chemical ecology project, you will study biologically significant questions, communication, defense, stress responses. You can go collecting in exciting places, hiking or diving trips, and potentially you can have high impact publications, though these projects do tend to be a little more difficult as well. So possible projects can encompass multiple themes. We could look at the semi-synthesis of cytochrome P450 inhibitors, so that's drug discovery and chemical ecology. We could look at metabolites mediating microbial interactions, which is drug discovery and metabolomics. We could look at metabolomic strategies to increase the efficiency of drug discovery,